Now we're going to move on to our next and last speaker for the session. Dr. Matthew Young is the program director at the Cancer Biomarker Research Group, Division of Cancer Prevention at the NCI. He's here to speak to us about his expertise and insight into early detection approaches for the family of GI cancers. Thank you.
Thank you very much, and I also like to thank the organizers for inviting me. This has definitely been an educational learning curve for me, so this should be good. So as mentioned, so this is what I'll talk about today. I'll talk about the Early Detection Research Network. I'll talk about some of our GI cancer consortia that we have up and running. I'll give you an example of biomarker development, specifically the Cologuard, and then we'll look into some multi-cancer screening with the liquid biopsy approach that's taking storm now, and then if there's time, we'll talk a little bit about some funding opportunities. So the Cancer Biomarker Research Groups, we do research, or we fund or promote research to identify and develop and validate biomarkers for early detection of cancer and risk assessments. And in this group, my portfolio includes the GI cancers, esophageal, gastric, and colon, and the pancreas. At this time, though, I'm not getting any funded grants in the gastric world, which they don't seem to get past the reviewers. Which suggests that we need an approach to get the reviewers educated as well on what needs to be done in gastric cancer. So, as you've heard many times, early cancer detection will save lives. If we're diagnosing cancers when they're large, most likely they've metastasized, and it's hard to treat or to intervene. So to reduce the mortality, we need to find effective biomarkers for early risk assessment, for early detection, and for early intervention studies. So that's where we go. That's what we do. And this slide is to show our approach to funding opportunities as the difference between working in an individual grant or a silo versus working in an integrated collaborative environment. So in your individual grants, you often will see many biomarkers reported. They're often unrealistic performance. They do great, 100% specificity, but they're hard to replicate in other labs. And they often use samples of convenience, what they have in the freezer. But if you form an integrated or a collaborative structure, much like we heard from Andy, that now the reported number of biomarkers are going to be less because many of the bad markers are going to be down-selected by the group. You have the performance is going to be more realistic. And they will perform better in multiple platforms or multiple cohorts. You can collect larger numbers of samples to perform these studies, and then you can the samples can be collected. Because the samples are collected at multiple sites, you really reduce the bias for your analysis side. So we believe that these collaborative structures are a much better approach for looking at biomarkers. And so I'm going to have some excerpts from Graham Ligon from Exact. He presented at the EDRN meeting, and so I've adopted some of his, in this case, lessons learned from doing biomarker studies. So case control samples are not the same as prospective samples. So in the late stages of a biomarker study, you really need prospective collected samples. You never have enough samples. That's all obvious. You need to understand where your samples are coming from and how they were collected and how they were stored. So these are critical things that cause failures in a biomarker validation studies. If the biomarker is actually a marker of the tumor or of the cancer progression, it's going to be more of a quantitative marker versus a qualitative, which is always a better component to the things. So, and too many markers in a panel is going to cause overfitting. So these are things that we have to consider when you work on, especially at the back and the end stages of doing these validation studies. So the EDRN was established over 20 years ago, and its focus is on biomarker research for early detection. We've set up guidelines for validation studies for collecting reference material. We actually have reference materials that we can make available to others that want to test out their markers or validate their markers. There's an unparalleled, fully operational access network. So most of what's been done through the EDRN is online and available for most people to look at and use in their own work. So the structure of the EDRN is a multi-lab, multifunctional assay program. It starts with it includes the biomarker development labs, the discovery labs for identifying new markers, reference laboratories that develop assays, mostly CLIO-specific type assays, and then a clinical validation center that will collect the samples in a blinded format that can be used for validating the markers as they come down the pipeline. The critical component of it is the data management and coordinating center. They maintain the blind on the samples. They maintain the assay collection or the sample collection so that they're all collected in a very uniform, precise manner, so that when when markers are tested across platforms, they should be consistent. So this is the structure, and many programs have adapted this type of structure as they've developed their own setups. So the whole point for the EDRN is to build clinical resources. We have a multi-center biomarker validation studies. Most of the assays or all the assays will be developed in a CLIO lab so that they're ready to go into a clinical validation study. And the mechanisms for the biomarkers are always triaged ahead of time before we put them in our large validation studies with our precious samples, so that we're not going to be testing things that are not really strong and validated. And then in fact, I think we've been through a couple thousand markers that they've triaged away, because the more you have coming in, the more likely you're going to find one or two at the other end. We've got, I believe we're up to 6 FDA-approved markers that have come through the EDRN, and another 20 or so CLIO-approved assays that are out there from this endeavor. So the Division of Cancer Prevention, that's where we are. We have other GI-specific consortia. They're set up to predominantly do early detection of cancers. These are so we've just started a liver consortia for translating research and for early detection of liver cancer. We have 2 consortiums focused on pancreatic cancer. One is a joint program with the NIDDK, and the other one is our program for early detection. Assad talked earlier about the Barrett's esophageal consortia, the BetterNet, and that's run out of his program. And then, the EDRN has three GI-focused labs in that. And then the last consortia, the MCL, is otherwise known as the overdiagnosis consortia. It is set up to understand how biomarkers that are coming down the pipe can create overdiagnosis and how we can avoid that. So this is a critical element so by putting these together, we work well. So we heard yesterday a lot about what we can do at Congress. So the PCDC and the CPD-PC consortia, the Pancreatic Cancer Consortiums, are a direct consequence of the Recalcitrant Cancer Act. So this was proposed, this was mandated by Congress in 2014. And from that, the NCI's recommendation was to develop a relationship between pancreatic cancer and diabetes. And the second point was to do longitudinal screening programs, protocols, and to collect samples, develop molecular imaging and biomarkers for patients that are high at risk. So that's those two mandates from the congressional mandates and the NCI recommendations. We actually use those mandates to recommend to the NCI that we fund these consortiums. And so both of them have been funded for close to 5 years now, and we're looking in the renewal states of that. And our report back to Congress is we still need to keep these types of fundings available. So this is one approach to get NCI to say, yes, we need to put more funds into that. So the Pancreatic Detection Consortium is it is a project of researchers and clinicians, and the idea is to identify and test biomarkers, body fluids, imaging biomarkers, precursors for early detections to determine if the pancreatic cysts are likely to progress to cancer. Obviously, we're picking up more of the cysts now, and so we need to know, do we move forward or do we watch and see? Develop molecular and image-based approaches and conduct biomarker validation studies. And a key component of this is to establish a biorepository that can be shared with other people along the way. And this is a similar structure that we put into all of our consortia because this is what is really needed, especially in some of our more rare or harder to study diseases. So, can we develop biomarkers for early detection of gastric cancer? As an example, these are what's out there for colon cancer. And you all know these. They're pretty much the only FDA-approved biomarkers for screening that are well established and commercially available. Excuse me. So the hemoglobin test is sort of the beginning, but we now have the FIT, Cologuard, and these are fecal tests. The epi-procolin is a blood test. And so you can see they all have a fairly good specificity and an okay sensitivity. So we can use this. We can deal with this kind of specificity and sensitivity for biomarkers because these are actually biomarkers that lead into a colonoscopy. So, and a colonoscopy is, while it's an invasive study, it is the standard of care in the U.S. So the biomarkers are to say you need a colonoscopy or you don't need a colonoscopy. So for pancreatic cancer, these specificities won't work because the next step is surgery, and that's not an appropriate, the specificity has to be much, much higher. So for, again, this is Exact's choice to make a new biomarker, which was Cologuard. Why do we need it? Well, there's a clinical need. There's a choice between using a FIT or colonoscopy, and then they want to have a more non-invasive with a little bit of higher sensitivity. To develop this biomarker. You first have to develop the collection devices and the assay platform. You have to make this a robust assay that you can do millions of tests a year, because that's what you want to do if you're doing a general population screen. And then you want to test that platform with enough samples to say that it works, and they chose 600. Then, because this is a multi-biomarker assay, you have to develop your algorithm, and they set and lock their algorithm on 1,000 samples. So then when you take it into the clinic to do the actual trial, they needed 10,000 asymptomatic subjects. These were people that were going in for a colonoscopy that never had a colonoscopy or symptoms. And so they tested that. And at the time this was done, the incident rates for people going in for a first-time colonoscopy asymptomatics was about 0.7%. So that's where the 10,000 sample patients come from. So when we hear in Chile, the incident rates is 0.16. So you can see where you're going to need significantly more to do this type of study, but we got to start somewhere, right? This is the timeline. They actually presented it, they developed a product at the Mayo early in 2009, and they had FDA approval sometime in 2014. So it's not really a significant timeline, but the actual development of and testing and deciding on which marker to use was probably another 10 years on the early side, because you don't hear as much on those. So these are the markers. It's a combination of methylated DNA, the hemoglobin or the FIT assay, the KRAS mutation, and a BRAF mutation. And you can see as you add on each one of the markers, the sensitivity of the test goes up. But as you add on these markers, the specificity goes down. Okay, so the more markers you add, the lower your specificity is going to be. So that's why you have to find that happy number that makes the assay work. So biomarkers for early detection require a high sensitivity and a specificity. The high sensitivity is needed to reduce the false negative results. The high specificity is needed to reduce the false positive. For colon cancer, the colonoscopy is a gold standard, and as I said, these markers are going towards a colonoscopy. So and they were actually validated against a colonoscopy, so it all works well. A false positive would be a misdiagnosis, but likely, especially the ones that have FIT in it, these will be picked up in 2 to 4 cycles of the assay, which is less than the 10-year on the colonoscopy. A false positive would lead to an unnecessary colonoscopy, but that's the standard of care anyway, so that's not really a major concern. So now if we look at gastric cancer, the endoscopy would be the standard of care, at least in Asia. Biomarker discoveries and validations can be verified by endoscopy if we're getting a good signal. The false negatives would lead to a misdiagnosis, but if we don't have any standard of care now, everyone is a misdiagnosis, so we can tolerate the false negatives. The false positives will lead to a unnecessary endoscopy, but that's not necessarily a major problem. So in a sense, we can run with biomarkers that have the similar sensitivity and specificity as the colon cancer markers. So what else is out there? So there's this I borrowed from another, the Johns Hopkins group on the race for developing early detection tests. This is all about the liquid biopsies. So these are the companies that have a stake right now at liquid biopsies that are actually doing clinical trials to test their markers. And most of these are multi-cancer type markers. So they're going to detect multiple cancers at a time. This comes from GRAIL, They just talk about the concept that a multi-test is the low false positive rates are achieved because you have a higher number of specificities. If you have more cancers, you're going in the test, you're going to hit one of the cancers, so the specificity goes up. And then similar with the high detection rate, they're going to find some cancer in the marker. So the idea is that we can get these markers through the clinic because they're going to hit colon cancer, or they're going to hit breast cancer, or something else. But the more rare cancers are just going to carry along with it. And so they all will have some kind of tag to identify the site of the cancer. So that's a very important thing. So the GRAIL is using a methylated marker. Where they're actually testing 15,000 people. Their data was actually shown at the ASCO GI meeting in January, and you can find it on their web page as a press release. So that's what's going up. They're focusing right now on the GI markers, or on the GI cancers, which does include stomach. Their N numbers they're low, so I'm not going to show you the data. You can go look at it. I think it's a little low to be really projecting where they're going. But they do have stomach in this panel. CancerSeq from the Hopkins group also contains stomach. This was published in Science in 2018. I think this was the first one out. The CancerSeq uses the DNA mutations with proteins to identify the target sites. You can see stomach is responding fairly well, so that's a good indication. Their clinical trial now, though, is only focused on ovary and liver. So that, I think it's a liver, so that's where their high marks are, so as they push it through. But as I said, if their clinical trial pans out to be positive, then all the other ones come along as just a carryover, and we can work with them. So if you've got a stomach and you score positive, you get an endoscopy. If it's wrong, then someone just got an endoscopy. Yeah, so it's not a bad thought where to go. Okay, so the last area always interesting to this group is the funding opportunities. So I just put in a simple search in our funding websites for gastric cancer, and this is all that came up. There's just two plans in here, R01s and R21s. The first two are health disparities. That's kind of obvious. And these are not targeted to gastric cancer. They just have gastric cancer in the subject matter somewhere. So that's why they came up. The last two are recently promoted provocative questions, and this is an RFA for early onset cancers. And as I understand, there is a rise in early onsets in gastric cancer as well. So this would be an eligible level for that. The RFAs for this these are the RFA numbers, and you can plug them in and do a research. The first date for applying is the 30th of April, and then November 17th on the second one. More than happy to talk about that if you have questions on that, and pursue that any further. So the main feature of this one is we're looking at the concerns about increasing in cancers in young populations. So you have to be able to show that this is something that's on the rise if you want to approach this. So if you have any questions, feel free to, and I'm going to skip over that. So thank you.
Thank you very much.